in breast cancer, spanning more than 2 decades, with strong foundation in research and development of 2 key approved medicines. In a few moments, I'll ask Roger Dancy to share our development plans, which have the potential to expand significantly our portfolio in breast cancer and bring forward potential new breakthrough treatments for breast cancer. Breast cancer is the most common tumor type among women in the United States, with 1 in 8 women receiving a diagnosis during their lifetime. It caused an estimated 44,000 deaths in 2023 alone. While early breast cancer can often be cured, many patients progress to advanced metastatic disease, which remains incurable. We're very proud of having introduced to the world the first CDK4/6 inhibitor, Ibrance, which has been a transformative therapy for hormone receptor-positive human epidermal growth factor negative metastatic breast cancer. Since its approval in 2015, Ibrance has been a market-leading CDK4/6 inhibitor and has treated over 665,000 patients globally. We see significant opportunity in breast cancer, which currently is a $30 billion global market that is expected to reach $60 billion in 2030. We are confident that with our differentiated pipeline, deep expertise, and strong relationships with the global breast cancer community, we are poised to not only continue but strengthen our leadership in this space. I'll now invite Roger back to speak more about our development plans in breast cancer, including several potential near-term catalysts. Thank you.
Yeah, thanks very much, Chris. Again, I mean, this is an exciting opportunity for us. I mean, breast cancer, it has a huge impact on the world, and it's a key focus for us. So, yeah, we're committed to improving breast cancer outcomes wherever we can. So it is a collection of multiple diseases. So if you look at the pie chart on the left of the slide, you can appreciate that the hormone receptor positive HER2 negative subtype comprises around 70% of all breast cancer cases, so a very large fraction. And then HER2 positive, the sort of traditional version of HER2 positive anyway, and triple negative breast cancer comprise the remainder. So in hormone receptor positive HER2 negative breast cancer, beyond the approved product Ibrance, our pipeline actually includes 4 promising candidates, 4 promising molecules, including 2 that are already in phase 3 development. And now with the addition of Pfizer and several early stage ADCs, as you can see on the slide, we now have the opportunity potentially to address the other subtypes, not just limited to the hormone receptor positive group. So in this presentation, I'm going to focus the 2 late stage molecules for hormone receptor positive breast cancer, that's atirmociclib, our highly selective CDK4 inhibitor, and vepdegestrant, the only PROTAC estrogen receptor degrader that's currently in clinical development. I'll also be discussing Pfizer, this remarkable CNS penetrant HER2 TKI, where we actually have active trials in early lines of breast cancer beyond where Pfizer has already been studied. And the clinical stage ADC, I think you heard earlier from Tom around disitamab vedotin, and we had some discussion in the Q&A. The full DV plan is not necessarily being presented here, but clearly we are interested in HER2 positive tumors, whether it's breast cancer, gastric cancer, or other cancers beyond urothelial cancer. And Jeff Settleman in Discovery Research will talk about felmatotug vedotin, otherwise known as B7H4, along with our potential first-in-class KAT6 inhibitor and novel CDK inhibitor. So I hope you can appreciate on this slide, we bring lots of potential to the breast cancer space. So turning to our efforts for next-gen therapies for hormone receptor-positive breast cancer, while the combination of CDK4/6 inhibitors with hormone therapy is the gold standard of treatment for hormone receptor metastatic breast cancer, not all patients respond adequately, and most ultimately progress and receive systemic chemotherapy. So there is a need to improve upon the existing therapies. And with atirmociclib, we believe we have the potential to continue our leadership that we've established with Ibrance, now introducing a new cell cycle inhibitor backbone that we think offers more complete and continuous CDK4 target coverage with improved tolerability. And with vepdegestrant, which we are co-developing with Arvinas, our aim is established in the same conceptual way, a new standard of care endocrine therapy backbone. And that's based on data which I'll share, impressive activity for vepdegestrant, regardless of ESR mutation status. And that ESR mutant status actually is a common driver for resistance to current endocrine therapy. So it is important to note that we have comprehensive development plans and programs for both of these molecules, and we plan to study the entire treatment continuum. So let me walk you through some of the data that underpins our confidence in these molecules. So I'll begin with atirmociclib, and the prospect of creating a molecule that specifically targets CDK4 while sparing CDK6 has been a challenge that our medicinal chemists have been working towards for a number of years. And we now have it in atirmociclib. This is a molecule that can potentially maximize the efficacy through selective CDK4 targeting while minimizing toxicity by sparing CDK6 and other kinases. There are some important biochemical attributes of atirmociclib which potentially differentiates it from CDK4/6 inhibitors. In the upper half of this slide, you can see kinome screens which show the results from over 270 activity assays for the current CDK4/6 inhibitors and atirmociclib. Just to orient you, if you're not familiar with these, the red dots depict regions where the molecules are most active, and the boxes focus on where the CDK4 and CDK6 domains reside. In the atirmociclib tree, all the way on the right of the slide, you can see almost exclusive activity in the CDK4/6 region with no meaningful off-target activity elsewhere. And with regard to selectivity for CDK4, if you look in the box below the kinome trees, atirmociclib has the highest selectivity amongst these molecules with a 33-fold ratio favoring CDK4 over CDK6. So essentially, this is a CDK4 inhibitor. So there's lots of preclinical evidence to suggest that the clinical data could point in the direction of better efficacy and potentially better tolerability. And I'd like to walk you through this data, which is updated from a phase 1 study evaluating atirmociclib in combination with endocrine therapy. And it's important to note, as with often these initial studies, patients in the study had received a median of 4 prior lines of therapy. 100% had received a prior CDK4/6 inhibitor, and most having received prior chemotherapy and prior fulvestrant. The overall response rate was an impressive 32%, and this is in a population where the benchmark would be less than 10% with available endocrine-based therapies. And if you look at the Kaplan-Meier curve on the right of the slide, out of this data set, the median progression-free survival observed at the time of data cutoff was 8.1 months. Again, in an indirect way, but it's the only way we can with single-arm data, the benchmark would generally be under 4 months. So this is impressive initial data. So not just the efficacy, but safety and tolerability, atirmociclib has the potential to differentiate. There have been no head-to-head studies between atirmociclib and CDK4/6 inhibitors, but the available safety and tolerability data points to where atirmociclib may offer advantages. Starting with neutropenia, as you can see on the slide, consistent with its selectivity for CDK4 and not hitting CDK6, atirmociclib plus fulvestrant appears to cause a lower incidence of neutropenia, including the higher grades where things potentially can go wrong for patients. So that's an important potential differentiator, and the numbers are quite different. With regard to GI side effects, atirmociclib also appears to have a low incidence of severe diarrhea, and that's basically because it's not a multi-kinase. It hits a very restricted kinase panel. And so probably most importantly, most notably, as a measure of tolerability, we see very few dose reductions due to adverse events, and with a low single-digit rate of discontinuation. I can tell you, just as been developing drugs for a long time, getting low single digits, discon, means a highly tolerable drug that can be used potentially for prolonged periods of time. And while these numbers are limited in terms of how many patients have we studied, the data does support the potential for atirmociclib to be dosed in that more complete and continuous fashion, which is critical in a disease state where prolonged therapy actually is required, because fortunately, the timelines for patients with breast cancer are long before something potentially goes wrong. So based on these data, we are working to develop this molecule across both early and late hormone receptor treatment settings. So starting from the left, we are currently enrolling a phase 3 registrational study, which is evaluating atirmociclib plus fulvestrant compared to physician's choice of fulvestrant or everolimus plus exemestane in the metastatic post-CDK4/6 setting. This trial is anticipated to read out in late 25 or early 26. So we've begun this journey in that post-CDK4/6 setting. In parallel, we are planning to execute a frontline phase 3 registrational trial, which will evaluate atirmociclib plus an aromatase inhibitor versus physician's choice of CDK4/6 plus an aromatase inhibitor as well. And obviously, this is at a very high level what we are thinking of doing, and more details to come as we get closer to that. But we look forward to initiating this study in the second half of this year. And in parallel, we are also advancing atirmociclib for potential use in early breast cancer, and we plan to initiate a signal-seeking trial in the second half of 2024. We will combine atirmociclib with an aromatase inhibitor, essentially a window of opportunity type of study. We believe that the development program that I presented to you has the potential to position atirmociclib as that replacement for CDK4/6 inhibitors across all lines of hormone receptor breast cancer settings and lines of therapy. Let's turn now to vepdegestrant, which is the first PROTAC ER degrader in clinical development. PROTAC, by the way, stands for proteolysis targeting chimeras and is a unique mechanism of action compared to other ER-targeted therapies. Vepdegestrant degrades and removes both the wild-type and mutant estrogen receptors to effectively interrupt signaling. And as mentioned earlier, ER mutations are actually a common cause of acquired resistance to endocrine therapy. Though I'm not sharing this with you today, we've been very pleased with the clinical data generated for vepdegestrant as monotherapy in heavily pretreated patients. But I will share with you some of the investigational combination data where we've combined vepdegestrant plus Ibrance, and we have demonstrated compelling anti-tumor activity in heavily pretreated patients with locally advanced or metastatic ER-positive, HER2-negative breast cancer. Shown here on the next slide are the interim data presented at the San Antonio Breast Cancer Symposium just a couple of months ago. And the population study is heavily pretreated, median of 4 lines of prior therapy, 87% receiving prior CDK4/6 inhibitors. And if you look at the waterfall plot on the left, you can see most patients in this circumstance combining vepdeg with Ibrance experience tumor shrinkage. So in this initial evaluation, the overall objective response rate was 42%, in a population, again, where response rates would generally be considered to be less than 10% with the standard endocrine-based therapies. and then looking at the outcome by the ESR1 mutation status, we basically see comparable response rates in both the ESR1 mutant and wild-type tumors, which is also very encouraging as a potential added attribute of this molecule. And then looking at the Kaplan-Meier curve, we are equally encouraged by the median PFS of 11 months, where again, the benchmark is generally less than a 4-month period with existing therapies. So vepdegestrant and Ibrance also demonstrated initial manageable safety and tolerability, but we are continuing to optimize the Ibrance dose for use in this combination. And if you look at the chart on the left, the treatment-related adverse events observed were consistent with the known safety profile of Ibrance in combination with endocrine therapy and the safety profile observed in studies to date with vepdeg monotherapy. Neutropenia was observed frequently in this combination study, likely related to higher than expected exposure to Ibrance, but was nevertheless still effectively managed through standard Ibrance dose reductions. And importantly, the dose-reduced Ibrance population in this combination was associated with durable responses with a median time on treatment of 36 weeks once the dose was reduced. So I think overall these combination results, as well as the previously disclosed vepdegestrant monotherapy data, have given us confidence to move forward with this molecule with multiple development strategies. Our intention, again, is to develop vepdegestrant as the backbone ER-targeting therapy, including in frontline and early breast cancer settings, and we have a number of ongoing and planned trials to support this approach. Let me begin on the left with registration intent studies in the second-line metastatic setting. First, we have the ongoing phase 3 VERITAC-2 study, which is evaluating vepdegestrant as a monotherapy, sufficient activity to evaluate as a monotherapy versus fulvestrant, with an anticipated readout in the second half of 2024, so pretty proximal to today. And subject obviously to clinical trial and regulatory success, we could anticipate that monotherapy would be the market entrant indication and potentially establishing vepdegestrant as that best-in-class novel endocrine therapy in late-line disease. And given the importance of combination therapy in more advanced aggressive disease, we are also planning a registrational study with vepdegestrant plus a CDK4/6 inhibitor versus standard of care in the second-line post-CDK4/6 setting. And we anticipate starting this study in 2025. In frontline metastatic breast cancer, we aim to establish this new backbone endocrine therapy concept. And as mentioned earlier, we are optimizing the dose of Ibrance in combination with vepdegestrant to manage things like the neutropenia. And that's in a study lead-in for the VERITAC-3 trial, which is also currently enrolling. And in parallel, we are evaluating the safety and tolerability of a combination of vepdegestrant and atirmociclib, the two novel agents, in a phase 1b study that is also enrolling. So data from this VERITAC-3 trial, the safety lead-in combination with Ibrance, data from the phase 1b/2 study of vepdegestrant in combination with atirmociclib will continue to inform the precise registrational path for vepdegestrant in the first-line metastatic setting. And we anticipate making a data-driven decision this year and initiating a phase 3 trial in 2025. Finally, we have an ongoing phase 2 signal-seeking trial of vepdegestrant versus AI in the neoadjuvant setting, and that too has an anticipated readout in 2024. So moving now on to Pfizer, our best-in-class tyrosine TKI for HER2-positive breast cancer. As we know, HER2-positive tumors are biologically more aggressive and are associated with poor survival compared with HER2-negative cancers. And one of the hallmarks of HER2-positive breast cancer is the development of brain metastases in upwards of 50% of patients with the condition over the course of their disease. And owing to its high selectivity for HER2 and remarkable brain penetrance, Pfizer has demonstrated an overall survival benefit and strong CNS activity in HER2-positive breast cancer. Shown here are the efficacy results from the pivotal HER2-CLIMB trial, which was unique in that nearly half the patients enrolled had brain mets, including those with active disease. HER2-CLIMB evaluated Pfizer in combination with trastuzumab and capecitabine in patients who'd received prior treatment with trastuzumab, pertuzumab, and T-DM1. Focusing first on the chart on the left, you can appreciate that the statistically significant and meaningful survival benefit with Pfizer, with the risk of death reduced by 34%. Looking at the Kaplan-Meier curve on the right, there was a clear benefit for patients with brain metastases, with a 52% reduction in the risk of progression or death. HER2-CLIMB also demonstrated favorable safety, low discon rates, and no requirement for prophylactic antidiarrheal medication, which is commonly used with less selective HER2 TKIs. So based on these results, we believe Pfizer is a best-in-class TKI therapy for patients with HER2-positive breast cancer, and we aim to extend its benefits to earlier lines of treatment. We're continuing to develop Pfizer as a backbone TKI in HER2-positive breast cancer and are particularly excited by the potential opportunity to delay or even prevent the development of brain metastases in earlier lines of therapy. HER2-CLIMB supported the initial approval in 2020 in that second-line plus metastatic setting, and more recently, HER2-CLIMB-02, which is also presented at San Antonio, demonstrated that you could combine Pfizer with an ADC, trastuzumab emtansine, or T-DM1, and improve PFS in the second-line plus metastatic setting. A final OS readout is anticipated sometime in the future, late 27 or early 28, to complete the dataset for that trial, but the results were very encouraging. So moving now to the HER2-CLIMB-05 trial, this is where we seek to establish this new frontline maintenance approach or backbone for HER2-positive metastatic breast cancer. And this trial is evaluating Tukysa plus pertuzumab and trastuzumab versus pertuzumab and trastuzumab, so it's an add-on design, after the completion of induction therapy, and we anticipate this readout in 2025. Finally, we aim to advance Tukysa in combination with TDM1 into the curative intent post-neoadjuvant setting, and this is being studied through a US cooperative group. The Alliance is running the trial, and that has an anticipated readout in 2028. So as you can appreciate, we are meaningfully expanding our portfolio in breast cancer, 8 compounds having active development programs, 7 ongoing phase 3 studies. We anticipate multiple key catalysts through the end of this year and into the first half of 25, with the potential for continuous readouts and additional data-driven opportunities spanning over the next 5 years. In the near term, we have the 2 phase 3 starts for atirmociclib, which I walked you through, with our second-line study in combination with fulvestrant already enrolling, and our frontline study in combination with an AI initiating in the second half of this year. We also look forward to readouts for Ibrance in the HER2-positive setting and for vepdegestrant as monotherapy. And in the second half of 25, we expect a readout from Tukysa, the HER2CLIMB-05 trial, and pending data from our ongoing combination studies with vepdegestrant, we will potentially initiate a phase 3 frontline study of the 2 novel molecules together, such as vepdegestrant plus atirmociclib. Longer term, we anticipate readouts from additional registrational trials for Tukysa, the frontline study for vepdegestrant plus either atirmociclib or Ibrance, and our 2 atirmociclib trials. Finally, we are excited by numerous opportunities across our expanding breast cancer portfolio, including the early stage clinical molecules that you heard about today. With our innovative pipeline, which spans many segments of breast cancer, we believe we are well positioned to reach a significant portion of the over 200,000 addressable patients in the US in the next decade. So thank you, and I'll turn it now back to Chris to discuss our plans in hematology oncology.
I was just not feeling my normal self. A series of doctors came in and they said something that was just foreign. You have multiple myeloma.
My first reaction when I found out was just devastation.
Multiple myeloma is a blood cancer. It impacts each individual patient differently. Treatment is an ongoing process. Those myeloma cells, they change up the script and they force you to have to be adaptable. It's not the end. It's the beginning of a new normal. I'm going to continue to live my life. I am a member of Pfizer's multiple myeloma patient steering committee. It's opened up a whole new world, sitting down with doctors and practitioners and giving them an interesting but different perspective from the vantage point of a myeloma patient. I'm very fortunate to be in a situation where health-wise I can still contribute. I am too busy to allow multiple myeloma to slow me down.
Thank you. I'm now going to walk you through our hematology oncology portfolio, including our significant opportunity with Elrexfio, our BCMA bispecific antibody for multiple myeloma. Pfizer has significant experience in hematology oncology with 9 approved medicines that reached over 22,000 patients with our combined portfolio in 2023 alone. These medicines span all 3 core modalities to treat various types of blood cancers, including multiple myeloma, chronic and acute leukemia, as well as lymphoma. With the addition of Adcetris, we now have an established blockbuster in our portfolio that is the foundation of care for CD30-expressing lymphomas. Adcetris is a first-in-class ADC that first received FDA approval in 2011. Today, it is approved in a total of 7 indications to treat Hodgkin's disease and certain T-cell lymphomas in various settings, including as frontline therapy for both adult and pediatric patients. We have a substantial near and long-term growth opportunity with our hematology oncology portfolio, most notably with Elrexfio, our BCMA CD3-directed bispecific antibody. We received the first FDA approval for Elrexfio in 2023 in relapsed or refractory multiple myeloma, and we have a comprehensive clinical development program ongoing to potentially expand its use across treatment settings. Our vision for Elrexfio will be the main subject of this presentation today. We are also exploring various novel and differentiated molecules in our clinical pipeline across modalities, including a potential next-generation Adcetris, and Jeff Settleman will cover some of these during his presentation. With the addition of the Seagen hematology team, we have substantially strengthened our capabilities with expanded experience and scale to benefit our medical and commercial efforts with Elrexfio now and in the future. As I noted, Adcetris has been on the market for more than 12 years, and it has been prescribed to over 55,000 patients in the United States. Our colleagues from Legacy Seagen have built deep relationships with the hematology oncology community across stakeholders, including physicians, patients, and patient advocacy groups. As part of the integration planning, we have cross-trained our customer field force from both companies. We have now doubled the number of commercial sales team promoting Elrexfio in the field. Importantly, we also now have integrated field medical teams delivering world-class healthcare provider scientific engagement, both in academic and in the community settings. This puts us in a solid position to broaden our presence with hem-onc customers as we launch the potential larger indications for Elrexfio, pending clinical trial and regulatory success. Now turning our attention to our opportunity with Elrexfio in multiple myeloma. There were approximately 35,000 new cases of multiple myeloma in the US in 2023 and approximately 13,000 deaths. Patients often cycle through several rounds of therapy, and after each relapse, there is a higher risk of treatment resistance, shorter remission, and shorter duration of response. That's why several companies are actively exploring many types of interventions and novel therapies for multiple myeloma. The global market size is significant today at $29 billion and forecasted to grow to $44 billion by 2030. Now, let me walk you through why we believe Elrexfio is well positioned to become a treatment of choice for multiple myeloma across the treatment paradigm. First and most importantly are the data. Based on the results so far, Elrexfio has a very promising efficacy profile with strong objective responses and complete remission rates, and remarkably long median progression-free survival in a highly refractory patient population with a median of 5 prior lines of therapy. Importantly, as a subcutaneous option, it has several convenience factors: flat, non-weight-based dosing, and a flexible dosing regimen. Elrexfio is the only BCMA bispecific with the ability to switch from weekly to bi-weekly as early as 24 weeks. It also has a tolerable safety profile with limited hospitalization during the step-up dosing period to manage the risk of cytokine release syndrome. In line with the data, the US label recommends that patients be hospitalized for 48 hours following the first step-up dose and 24 hours following the second step-up dose. This is half the total step-up hospitalization time recommended in the US label for other approved BCMA bispecific antibodies. Now let me go into more detail about the efficacy data. The first pivotal trial for Elrexfio is MagnetisM-3, a phase 2 study in patients whose disease has relapsed or is refractory to 3 primary classes of existing therapies. This is a broad, very heavily pretreated patient population. In particular, I want to call your attention to the table on the left. A high percentage of patients, 97%, enrolled in MagnetisM-3 and were considered triple-class refractory. Also, approximately 40% of patients were penta-drug refractory, receiving up to 6 lines of previous therapy. In addition, MagnetisM-3 included a significantly higher number of patients with extramedullary disease, which is also an unfavorable prognostic marker. This is a very advanced patient population with characteristics associated with poorer outcomes. Despite this, Elrexfio has shown the longest reported median progression-free survival at 17.2 months for this setting. Also of note, the Elrexfio curve appears to have plateaued around the time of the median. While we can't draw conclusions with cross-trial comparisons, we are very encouraged by these data. If we now focus in on duration of responses with Elrexfio, we can also see the plateau of the Kaplan-Meier curves. On the left is the overall responding population, where the median duration of response has not yet been reached. On the right is the striking durability of response for the almost 40% of patients who achieved a complete remission or better. Median duration of response has still not yet been reached at 18 months follow-up. And again, you can see the plateauing of the curves. We believe these are truly remarkable outcomes in this setting. These are very deep and durable responses. And we now have patients who've been treated with Elrexfio for more than 3 years in this very late-line setting. We also plan to update these data, including overall survival data from the MagnetisM-3 study later in 2024, which we believe has the potential to solidify the compelling clinical benefit of Elrexfio. We expect these data to only get stronger as we move to earlier lines of therapy. We have designed a well-thought-out development program to offer an Elrexfio-based treatment regimen across all lines of therapy. And over time, we anticipate expanding to significantly larger eligible patient populations with longer durations of therapy. In the relapse refractory setting, the eligible patient population is 18,000 in the US. We have 2 studies ongoing here, MagnetisM-5 and MagnetisM-32. MagnetisM-5 has been designed to evaluate the superiority over a daratumumab-based regimen in the second-line plus setting. This study has completed enrollment, and we anticipate results in the first part of 2025. MagnetisM-32 is a trial designed to address unmet needs in the rapidly changing landscape to position Elrexfio as a potential new standard of care for patients with disease relapsing on anti-CD38 and immunomodulatory therapy in the frontline setting. Our goal here is to generate data establishing Elrexfio as the go-to second-line choice in the relapse refractory setting in the medium term. In parallel, we're evaluating Elrexfio in the newly diagnosed setting, where the patient population significantly expands to 31,000 in the US. We have 2 ongoing studies here, the MagnetisM-7 trial as a post-transplant maintenance option and the MagnetisM-6 trial in transplant-ineligible patients comparing Elrexfio to the current standard of care. These studies are recruiting well, with results expected in 2027-2028 timeframe. Altogether, these 4 studies are enrolling more than 2,000 patients over the next few years, which speaks to the strength of the data we have to date and the strength of the data generated with Elrexfio. In summary, we are excited by the potential for Elrexfio to reach a rapidly expanding number of patients globally, subject to approval and positive readouts of ongoing studies. We are really encouraged by the data generated so far with a remarkable long median PFS and a deep and durable responses in the very late-line setting. There are multiple catalysts anticipated over the next 2 years, including data from the MagnetisM-5 and MagnetisM-32 studies and the second-line plus setting. And longer term, we anticipate results from the additional pivotal trials to potentially move into the frontline setting. In addition to these pivotal programs, we also are investigating potentially differentiated novel combination approaches with Elrexfio, as well as the early disease setting with a phase 2 study ongoing in smoldering multiple myeloma, collaborating with the European Myeloma Network. With our ongoing phase 3 program, we are well positioned for Elrexfio to reach a significant portion of the over 50,000 addressable patients in the US during the next decade. We also have additional candidates in development in the early pipeline. We are planning to start phase 1 and 2 studies with additional mechanisms of action for various types of blood cancers through 2025 and anticipate providing more updates of these in the next 1 or 2 years. Thank you, and now I'm going to switch gears to our next generation opportunity presentation by Jeff Settleman. I'd like to introduce Jeff Settleman, our Chief Scientific Officer. Jeff leads all oncology research from the beginning stages of discovery to IND-ready programs. Jeff is an internationally recognized leader in molecular targeted cancer therapeutics, in the epigenetics of cancer drug resistance and personalized cancer medicine. He held numerous notable positions in both academic research and drug discovery before joining the Pfizer team. Prior to his current role, Jeff served as head of oncology research at Calico Life Sciences, and prior to Calico, he led discovery oncology at Genentech. I'll now turn it over to Jeff. Thank you.
Chris. So thank you all for joining us today. You've now heard about several of our approved cancer medicines and several advanced clinical stage programs. And now I'm excited to discuss our current areas of research focus and a few potentially transformative early stage programs. So I'll start with this slide, which simply highlights our 2 major R&D hubs for oncology. Both are on the West Coast. One is the newly acquired site in Bothell, Washington, north of Seattle, formerly a Seagen site. And the other is in La Jolla, where we recently made the decision to move to a brand new state-of-the-art facility, pictured here. It's located a few minutes from our current site. And the vast majority of our lab-based oncology research activities will take place at these 2 sites. And we're already seeing how having our two main research sites in the same time zone can actually go a long way toward facilitating communication and collaboration. So with the acquisition of Seagen, we see many opportunities to combine our scientific expertise, our technologies, and our molecules to enhance the discovery and development of the next generation of cancer therapies. And here we're highlighting 3 especially important opportunities that we see for scientific synergy. First, we'll be leveraging Pfizer's protein engineering and antibody design capabilities to enhance the legacy Seagen's ADC technology to deliver next-generation antibody conjugates with innovative new features. We'll also be incorporating Pfizer's deep expertise in small molecule drug discovery into legacy Seagen's ADC platform to advance next-generation ADCs with differentiated payloads with new mechanisms of action. And lastly, we'll be combining legacy Seagen and Pfizer molecules from our now substantially expanded aggregate portfolio to enable new mechanistic synergies and potentially reach many more patients. And with our expanded portfolio, we have new opportunities to attack tumors from multiple angles and to extend the reach of our science in many indications. The portfolio currently features 3 modalities enabled by deep technical expertise and experience. Those 3 core modalities include the ADCs, as you've heard about, the IO biologics, including bispecific antibodies, and the small molecules. Our ADC portfolio grew substantially, of course, with the acquisition of Seagen, and this platform has already successfully enabled 6 of the 11 FDA-approved ADCs. And the next generation platform is now aimed at novel targets and improved and differentiated payloads. Several of them are listed on the slide here, and I'll focus on the ones in blue today. Moving to the IO biologics, you heard about Elrexfio from Chris just now, and we continue to leverage our combined expertise in protein engineering and antibody design, as well as immuno-oncology to develop next-generation biologics. And in the small molecule space, Pfizer has a long history of deploying world-class structure-guided drug discovery and medicinal chemistry expertise to deliver innovative, best-in-class and first-in-class small molecule cancer medicines. Targets for some of these are listed here on the slide, and they include some approved medicines, clinical candidates, and there's one preclinical program that's expected to enter clinical development very soon. I'll be discussing the ones highlighted in blue here. And all of these were discovered or enabled by work done in Pfizer research labs. In addition to the clinical stage programs, we have a discovery and preclinical stage pipeline with 63 programs currently, with opportunities for rational combination therapies with the potential to change treatment paradigms in many indications. And now I'll spend a few minutes on each of these 3 modalities, starting with next-generation ADCs. So with this timeline of ADC development, it's easy to see that Pfizer now has a substantial footprint on the current landscape of ADCs. Currently, there are 11 FDA-approved ADCs, 5 are Pfizer products, 2 others employ licensed Seagen technology, and 1 additional Pfizer product listed here, dasatinib vedotin, which you heard about earlier, is approved in China. And now we're looking to build on that success with next-generation ADC technology, which I'll describe. So to further strengthen our leadership in the ADC space, we're advancing our pipeline with novel targets and diversified linkers and payloads. So starting on the left, I've listed 4 of the FDA-approved vedotin ADCs to highlight the strength of the vedotin linker payload. And this is why we're still developing vedotin ADCs, as you've heard. Moving to the right, we've listed clinical stage vedotin ADCs. You heard about the encouraging efficacy already for 3 of these, and I'll have more to say about the felmetatug, or the B7H4 ADC. As Megan noted, the vedotin ADCs can elicit an especially immunogenic cell death with the potential for robust combination efficacy with immune checkpoint inhibition. This was very nicely exemplified by the Padcev-Keytruda combination clinical experience, as Tom described earlier, with synergistic efficacy seen in urothelial cancer. And then moving again to the right, we also recognize the important opportunity to complement the vedotin technology with some novel differentiated linkers and payloads. Here we've listed several programs that employ a novel, very potent topoisomerase inhibitor payload, and I'll describe 2 of those shortly. Moving again to the right, we're also developing next-generation auristatin payloads with potentially improved tolerability. And then moving all the way to the right, you can see where we're describing some discovery and preclinical stage programs that employ payloads with distinct mechanisms of action, for example, immune agonists, and I'll discuss the first one listed there. And then we're also pursuing protein degraders as antibody conjugates, both with internal capabilities and through a research collaboration with Nurix, a leading protein degrader company. And we're especially enthusiastic about this protein degrader class of payloads. And we're pursuing various novel cytotoxic payloads with some very highly differentiated mechanisms of action. So with most of these programs now either in the clinic or moving to the clinic this year, we believe we're now uniquely positioned to bring forward the most extensive ADC portfolio in the industry, which includes a variety of novel and differentiated programs. Now I'll go deeper on a few of the next generation ADC programs, and I'll start with CEACAM5C, which includes a potential best-in-class topoisomerase 1 inhibitor payload. This program is partnered with Sanofi, and it employs their CEACAM5-directed antibody, tusimidimab. So CEACAM5 is a protein expressed on the surface of many solid tumors at relatively high levels. I've listed some of the tumor types here. It has a drug-antibody ratio of 8, this particular ADC. And we're seeing some very remarkable activity in preclinical models, shown here in a panel of patient-derived xenografts from both lung and colorectal cancer, where we see very strong, robust tumor growth inhibition across many models. And this is notably following a single administered dose of the ADC. So it's a very promising program. The phase 1 dose escalation is now underway. Clinical proof of concept is planned initially in colorectal cancer with an opportunity to expand to additional solid tumor types. So on this next slide, I just showed you data that highlights the robust anti-tumor activity that our topo-1 inhibitor can deliver as a payload in preclinical models. Now here we're directly comparing our topo-1 inhibitor payload to the deruxtecan topo-1 inhibitor in preclinical tumor models. So there are 3 examples shown here with 3 different ADC antibodies. And what you can see in each case, where we've taken each antibody and conjugated it either to our topo-1, our novel payload, or to deruxtecan, labeled DXD here. The topo-1 inhibitor, ours is in the dark blue curve. And in all 3 cases, you can see evidence of a superior anti-tumor control with the matched antibody that includes the topo-1 novel inhibitor. So we're very optimistic about the potential for this new topo-1 inhibitor as a differentiated ADC payload that drives robust anti-tumor activity across many models. And we expect to have up to 4 ADCs with this new payload in the clinic by the end of next year. And one of those programs is described here, 35C. It's a next-generation CD30 targeted ADC with the potential for an improved therapeutic index versus brentuximab vedotin, also known as BV or Adcetris. This is our approved ADC targeting CD30. And this also includes our novel topo-1 drug linker. And in non-clinical lymphoma models, we've observed dose-dependent potent anti-tumor activity, as shown on the upper graph in a Hodgkin's lymphoma model. And then in a tumor model below that that's resistant to Adcetris due to increased drug efflux activity, this 35C ADC shows very robust anti-tumor activity, suggesting the potential to overcome clinical resistance to Adcetris that's associated with drug efflux, which could be very impactful for patients. We've also seen evidence of improved tolerability in preclinical models. So there's no observed hematologic toxicities at the highest non-severely toxic dose, and no pulmonary toxicity seen at any tested dose. So we're excited about this program, which is expected to enter phase 1 clinical development this year. And now I want to touch on our PD-L1 IT program, which involves a unique antibody conjugate approach to promoting anti-tumor immunity. So this molecule, which is illustrated here, is designed to deliver a potent immune-stimulating TLR7 agonist to the tumor microenvironment. Last year at AACR, we described a macrophage targeted antibody conjugated to this TLR7 agonist, which is shown on the upper right, that it induces much lower levels of systemic circulating cytokines, such as IL-6, which is shown here, when compared with administration of the unconjugated TLR7 agonist. And when we conjugate this TLR7 agonist to a PD-L1 antibody, we see potent single-agent tumor regression on the bottom graph, and this is maintained even following tumor rechallenge. So together, these data suggest that this antibody-conjugated TLR7 agonist has the potential to deliver a very effective dose of TLR7 agonism to the tumor microenvironment, but without the immune toxicity that one would expect with systemic administration of a TLR7 agonist. And this antibody conjugate uses that same proprietary PD-L1 antibody that Megan discussed earlier, which was designed for enhanced internalization, has an optimized drug linker and a payload and an antibody format that's been optimized as well. So we have very high hopes for this new immune-stimulating antibody conjugate. The IND is expected this year, and our target indications include non-small cell lung cancer, head and neck squamous cell cancer, and melanoma. So here on the next slide, we're highlighting early phase 1 data not previously shared with one of our investigational vedotin ADCs, felmetatug. The ADC targets B7H4, which is highly expressed in many tumor types, especially on breast, endometrial, and ovarian cancers, with much lower levels on normal tissues and immune cells. And we're seeing in these waterfall plots to the right very encouraging efficacy in our phase 1 dose escalation study from cohorts of triple-negative breast cancer and HR-positive, HER2-negative breast cancer. And as shown in the table below, there are early signs that B7-H4 levels could be a potential biomarker with an expression-response association in triple-negative breast cancer, such that the overall response rate in all comers was 21.4%, but it increased to 38% in patients with more than 50% of tumor cells expressing B7-H4. It's an early signal. It'll require more data before we can establish clearly whether this could be a biomarker used to stratify patients for treatment. And while we're only showing clinical activity here in breast cancer, we're also exploring endometrial and ovarian cancers, and we're very encouraged by early signals in all of these indications. So now I will pivot from our ADCs to our immuno-oncology biologics, which includes bispecific antibodies as well as other antibody-based programs. So with the acquisition of Seagen, we are significantly expanding our immuno-oncology biologics pipeline with a variety of innovative new programs, 2 of which are shown here. One of these on the left involves an immune engager bispecific antibody that was exclusively licensed from Lava Therapeutics. It's designed to redirect so-called gamma delta T cells to tumor cells to promote tumor cell killing, and I'll go deeper into that program in a minute. And then on the right is a second program that originates from our research labs in La Jolla and involves agonizing the lymphotoxin beta receptor to promote so-called tertiary lymphoid structures in tumors. And I'll explain the therapeutic hypothesis in a moment. So on the next slide is a description of our EGFR D2 bispecific program from Lava. It's a potential first-in-class gamma delta T cell redirecting treatment for solid tumors. One arm of this bispecific antibody binds to EGFR on the surface of tumor cells, and the other arm binds to the gamma delta class of T cells and activates and delivers those T cells to EGFR-expressing tumor cells, resulting in an innate immune response. And on the right, I'm showing survival curves from a preclinical xenograft model derived from a KRAS mutant colorectal cancer. And here, the bispecific antibody at 2 tested doses extends survival of tumor-bearing animals, so providing some nice preclinical proof of concept for this novel modality. And importantly, in exploratory preclinical safety studies with a surrogate antibody, we saw no cytokine release syndrome, suggesting the potential for less immune toxicity than we've observed with other types of T-cell redirecting immune engager antibodies. The phase 1 dose escalation study is now underway, and the key potential indications include colorectal cancer, non-small cell lung cancer, and head and neck squamous cell cancer. This next program involves a very novel therapeutic approach in which we've discovered an agonist antibody that engages the lymphotoxin beta receptor to induce so-called tertiary lymphoid structures, or TLSs. These are immune cell aggregates resembling secondary lymphoid organs. And they've been reported in several tumor types, and there are accumulating reports that show an association between TLS formation and maturation and response to anti-PD-1 therapy. An example of that is shown in the graphic on the left. It's from a Nature paper, which suggested that in melanoma patients treated with an anti-PD-1 antibody, that their overall survival was significantly increased as a function of having more mature TLSs in their tumors. So the hypothesis is that promoting these TLSs could have therapeutic benefit for some solid tumors, especially when combined with checkpoint inhibition. So to activate the lymphotoxin beta receptor, we discovered a potent antibody agonist pictured in the middle, which involves an innovative tetravalent antibody format that activates the receptor, leading to expression of multiple immune cell recruiting chemokines. And when we treat tumor-bearing animals for several weeks with this agonist antibody, we see evidence of treatment-induced TLSs, as shown on the right in these pre- and post-treatment tumor tissue sections stained for markers of TLS structures, showing that this antibody has the potential to induce the formation or maturation of TLSs, at least in a preclinical tumor model. So this unique and very exciting and differentiated IO program is expected to enter the clinic in the next few months. Now I'll move on to the last of our 3 core modalities, our next generation small molecule inhibitors. So I'll start with our first-in-class CDK2 selective small molecule inhibitor, which is designed to prevent or overcome resistance to the widely used CDK4/6 inhibitors in ER-positive breast cancer. And we see this molecule as a potentially important combination partner for our CDK4 selective inhibitor, which Roger described earlier. Now, CDK2 and CDK4, as shown in the schematic on the left, can cooperate to regulate some understood key cell cycle checkpoints, suggesting some potential functional redundancy between CDK2 and 4. And consistent with that hypothesis, in preclinical and clinical studies, we've seen evidence that activation of CDK2 may play a significant role in resistance to CDK4/6 inhibitors in a subset of breast cancers. So we've hypothesized that combining CDK2 plus CDK4 inhibitors could deliver synergistic benefit and potentially prevent or overcome resistance to the standard of care treatment. Here are some supportive non-clinical data in the form of xenograft studies from 2 different models. The 2 on the left are from a CDK4/6 naive model, and on the right are from a palbociclib-resistant PDX model, patient-derived xenograft model. There are monotherapy controls, but also combination treatment arms in each case, either CDK2 plus palbociclib on the left, followed by CDK2 plus CDK4 inhibitor, the same in both pairs. 2 key takeaway here. We see clear mechanistic synergy with the CDK2 plus 4 combination, so the second and fourth graphs. The purple curves are the 2 plus 4 treatment. We also see that CDK2 does better when combined with CDK4 than with palbo, than CDK2 plus palbo. That is in the red on the first and third graphs. So this is not surprisingly, we know, as you heard, that CDK4, our CDK4 inhibitor, is predicted to cover CDK4 much more effectively than palbociclib can, and also spares CDK6, and therefore has the improved safety profile that you saw earlier. Now, in phase 1, we've seen monotherapy efficacy with confirmed PRs in heavily pretreated ER-positive, HER2-negative metastatic breast cancer patients. This was reported at ASCO. And we have multiple ongoing dose expansion cohorts underway, CDK2 inhibitor plus endocrine therapy, and then a CDK2 inhibitor plus endocrine therapy, plus our CDK4 inhibitor. So we're excited to build on the encouraging clinical efficacy that Roger described for our CDK4 selective inhibitor with this rational, scientifically understood combination approach. Next, I want to discuss our next-generation brain-penetrant mutant BRAF inhibitor, which is designed to address some of the limitations of currently approved BRAF inhibitors. So to remind you, on the left, 39% of melanoma patients harbor BRAF mutations, and the activity profile of our inhibitor preclinically predicts that it should inhibit several forms of mutant BRAF proteins. There's the class 1 that are most commonly seen on the top there, and as well as class 2 and class 3, which are not addressed with current BRAF inhibitors. We predict that our new BRAF inhibitor will cover these mutations, as well as mutations that importantly arise on treatment with current BRAF inhibitors and lead to the resistance associated with mechanisms shown below, such as BRAF splice variants and NRAS mutations. And importantly, these resistance mutations, which appear in the second row here, can potentially be addressed with our novel inhibitor. In fact, in non-clinical models, our molecule displays activity against several of these mechanisms shown here, including the BRAF splice variants and the activating NRAS mutations. And then on the right are some non-clinical experimental data showing superior anti-tumor control in a melanoma xenograft with that has a BRAF class 1 mutation when comparing our new BRAF inhibitor, brain-penetrant BRAF inhibitor, with a variety of things here, but most notably the encorafenib-binimetinib combination standard of care doublet, which is the purple hashed lines on the upper graph, versus on the right in blue, the single-agent treatment with this novel BRAF inhibitor. So very impressive comparative preclinical data. And then below that, as a brain-penetrant BRAF inhibitor, this molecule demonstrates superior anti-tumor control in an intracranial xenograft model when compared to the encorafenib-binimetinib doublet, which is on the left. You can see on the right, single-agent treatment with this new BRAF inhibitor, very remarkable anti-tumor activity in this intracranial model. This is underway. There's an ongoing phase 1 study where multiple confirmed responses in RAF inhibitory refractory patients have been observed both systemically and intracranially. These promising new non-clinical and clinical data will be presented at the AACR meeting in April. So lastly, I'll discuss our KAT6 inhibitor program. So KAT6 is an epigenetic regulatory enzyme, a histone acetyltransferase. The KAT6 proteins are part of a gene regulatory complex that affects the expression of genes involved in estrogen signaling, cell cycle signaling, oncogenic signaling, and we've discovered a very potent and selective first-in-class small molecule inhibitor of KAT6. In preclinical models, this molecule can potently repress the expression of the estrogen receptor that's shown in the upper right. There's brown immunohistochemistry staining from an estrogen receptor-positive xenograft of a breast cancer. And on the right, following treatment, 3 mg per kilogram, we see very strong repression of expression of the estrogen receptor. And then below that, we see dose-dependent tumor growth inhibition in a preclinical xenograft model derived from an ER-positive breast cancer patient who had been previously treated with palbociclib plus letrozole, where we see complete regression at 3 milligrams per kilogram of daily treatment. So a very potent molecule. And in a phase 1 study that's underway, we previously reported several confirmed responses during dose escalation, and now we're showing for the first time some very promising data from an expansion cohort of metastatic ER-positive, HER2-negative breast cancer patients who had all progressed after CDK4/6 inhibitor plus endocrine therapy. 43 patients in this cohort, all heavily pretreated, as many as 6 prior lines of treatment. They were treated with 5 milligrams once per day of the KAT6 inhibitor plus fulvestrant. And we've seen very encouraging and durable clinical benefit. A confirmed overall response rate in this heavily pretreated patient population of 27.9%, and importantly, a median PFS of 7.5 months, which is 5 months improvement over fulvestrant alone in this setting. It's generally well-tolerated treatment, only 7% discontinuation rate due to AEs. Most frequently, we saw low-grade dysgeusia, change in taste, and we're now optimizing the dose and schedule to improve on this dysgeusia, and then we'll decide how best to advance this program, which is highly differentiated but very promising, into late-stage clinical development. And now, in a final slide, we've listed here many of our promising early clinical and preclinical programs, just to give you a broader perspective on our early oncology pipeline. Notably, we've only listed selected preclinical stage programs here. A few of these haven't been disclosed previously, and we have several others that we expect to be able to disclose in the near future. We also have a large number of discovery stage programs in our newly expanded oncology pipeline that aren't shown here. And so with this diversity and scope of innovative new molecules, we're very enthusiastic about the opportunity for our early pipeline to deliver multiple potentially transformative new medicines across many indications. So thank you, and I'd now like to turn it back to Chris.
Thank you, Jeff. You've heard a lot about our vision and about our portfolio. I would now like to ask Sunit Varma to come up and present to you how we will potentially bring these great and breakthrough therapies to patients.